The David Marsden Lecture was created to recognize an outstanding scholar and inspiring neuroscientist in the field of movement disorders. This year's awardee is Professor Olle Lindvall from the University of Lund, who perfectly fits this profile. Professor Lindvall received his PhD in 1974 and MD in 1978 from the University of Lund in Sweden and has been affiliated with the school for more than 45 years. He served as chairman of the Division of Neurology until 2012, the Department of Clinical Neuroscience from 2001 to 2003, and was vice dean of the Medical Faculty, University of Lund, 1997 to 99. And in 2014, he was appointed senior professor of neurology at the University of Lund. Would be impossible because of the innumerable achievements and contributions that Professor Lindvall has generated to enumerate all of them, but I would like to mention that he has led the clinical cell transplantation program for Parkinson's patients at Lund University Hospital between 1983 and 2012. And this program pioneered the use of neuronal replacement as a possible novel therapeutic strategy to restore function in the diseased human brain. And in that regard, Professor Lindvall will be talking about developing cell therapy for neurodegenerative disease, a lifelong journey. Professor Lindvall.
First, I would like to thank the Movement Disorder Society for selecting me to give this lecture. David Marsden was a great inspiration to me during an important part of my career. And every time I met him, as on this photograph from 1987, I felt that I got new energy in my work. David was definitely the most brilliant scientist clinician I have ever met. Trying to repair the brain and restore function in Parkinson's disease and other diseases by replacing dead cells with new healthy cells has really been a lifelong journey for me. And since you heard already yesterday about the current status of cell transplantation in Parkinson's disease, I thought that I would instead give you some personal recollections from this scientific journey, and end up by describing to you why I think we are in an extremely exciting time when it comes to cell therapy for brain diseases. When I started as a young physician in clinical neurology in 1976, after having finished my PhD thesis in 1974, the situation was very different. And the dogma of brain repair and plasticity is very well illustrated by Cajal's words. In the adult brain, the nerve paths are fixed and immutable. Everything can die, nothing can be regenerated. And when I was talking about some very exciting data that has come out from basic science in the mid or late 70s with my first boss, Ragnar Müller, who was an excellent clinician and lecturer, he just met me with strong disbelief. He said to me that the clinical application of brain repair has never been possible in the past and will never be possible in the future. But in 1979, there were two articles published which actually had clear clinical implications. And those two were published independently by the Stockholm group, together with people in the collaborators in the United States, and by the Lund group with Anders Björklund and Uns Steneve, showing that you could transplant fetal mesocephalic dopamine-rich tissue to the brain of rodents and ameliorate the symptoms of Parkinsonism in an animal model. And this, of course, triggered directly the possibility, or raised the possibility, that it would be possible to develop a transplantation therapy for patients with Parkinson's disease. I was very excited about these data, about the preclinical data, but I was very hesitant about the clinical translation for two main reasons. The first reason was the practical one. Would it be possible to collect human fetal mesencephalic dopamine-rich tissue from routine abortions? Would it be possible to identify the ventral mesencephalon containing the dopaminergic neurons? And finally, would it be possible to put cells into the human brain without any adverse effects? The second hesitation, the second concern was, of course, and most important, was the ethical one. Would it be morally and ethically justified to use tissue from aborted human fetuses, collect, and then transplant them to patients with severe motor problems, to Parkinson patients with severe motor problems? And this was not easy. I had, after a long, and took time, and after a long consideration, I came to the conclusion that it is, in my view, justified ethically and morally to use human fetal tissue as well as human embryonic stem cells for this purpose. But the first transplantation in the clinic were not performed with human fetal mesencephalic tissue, but with adrenal medulla tissue. And those studies were based on an article in Nature providing some evidence that grafts of adrenal medulla tissue containing, of course, catecholamines, that they could ameliorate a rotational asymmetry in a model of Parkinson's disease. And this led Erik Olof Backlund, Åke Seiger, and Lars Olsson to perform two implantations, or implantation in two patients in 1982 and 83 with implants in the caudate nucleus. And when I met with Lars Olsson at the meeting, we decided that we should start a collaboration and introduce two major improvements. The first one was to implant in the putamen instead of the caudate nucleus in order to have a better chance of improving motor function. And the other was to actually improve the neurological assessment of motor function, to be able to detect also changes in parts of the body. And so actually the data from the first patients were inconclusive. So we developed a test battery to detect changes therapeutically valuable or not in motor function in patients. And patients were, then two patients were operated on 19 and 20 of April 1985. And I remember this very, very well, because in the surgery room was crowded. There were more than 20 people who wanted to be present at this unique occasion. It was the third brain transplant in the world. And I remember we also had a syringe of haloperidol available in the surgery room in case the patient would react with an acute psychosis due to catecholamine release. But nothing happened, however. And the patients were followed very closely, and we made also over the first 6 months. And I was going to report about these data at a meeting at the New York Academy of Sciences. The data showed that there was a very slight transient improvement in motor function, primarily on the side contralateral to the graft. And I remember when I was describing the data, I gave a talk at this day, April 4th, 1986, at the New York Academy of Sciences. And I was met with, the audience was overtly negative to my talk. And people literally threw rotten tomatoes at me. And interestingly, the discussion was published. So even if the text is slightly polished, we can read statement like this by an excellent neuroanatomist, Constantino Sotelo. Having been a neurobiologist for 25 years, I would rather do experiments in animals than in humans. By a colleague, Mark Perlow, you describe the beneficial result to catecholamine release. I don't find any evidence in your paper that there was any catecholamine release, and I wonder how you could come to that conclusion. And then from Stan, who I admired tremendously already at that time, it always amazes me how careful basic laboratory scientists are to have controls in animal experiments. But once they start working on patients, the control disappears out of the window, and we are left with bad treatment for years. I was disappointed, and I was very worried, because in my view, the study had given some important information. First, that it was possible to put cells into the human putamen without adverse effects. And second, that we could detect some minor changes using our test battery. But would it be possible to continue with clinical transplantation, despite the fact that the recommendations of the meeting was to continue with animal experiments for the next 10, 15 years? That night I shared hotel room with Anders Björklund, my colleague. We had done a lot of studies during the 70s and 80s. This is an early photograph from Paris when he had a lecture tour in 1974. And we shared hotel room, and during the night we discussed this arguments for continuing with human fetal tissue or with cell transplantation, and we decided, I remember it very well, that we should continue with the cell transplantation program with human fetal tissue, despite the resistance of the scientific and clinical community. What we did then as one step, and a very important step, was a meeting with Richard Frakowiak and David Marsden in London. And the question at that meeting, which was held in June 1986, was, would the available methods have the resolution and sensitivity so it would be possible to detect surviving dopaminergic graft after transplantation in a patient's brain? And this had never been done. No one knew if that would be possible, but after 2 hours of discussion, we came to the conclusion that it might be possible using fluorodopa and PET scanning to detect a surviving dopaminergic graft. So in September 1987, I went to, together with my nurse, to London with 2 patients for PET scanning and preoperative PET scanning. And the patients needed a helmet in order to, so that there would be no movements during the PET scanning. And then also the patient had to lie down several hours during the PET scanning without having taken anti-Parkinsonian medication. And this was, of course, extremely tough for the patients, and they were really heroes. This shows the photograph when we have completed, or Nico Lenders, who had completed the first PET scanning, and everyone was very happy. Richard Frakowiak, who you can see to the left, he was opening a bottle of champagne, and we all cheered and were very happy about this successful scanning. Unfortunately, we lost most of the data during the night due to a computer failure, so Richard became furious. An important step in the clinical translation was this work which we had together with Patrick Brandin, and which was included in his thesis, where we could show that we could get survival of dopaminergic neurons from human fetuses in rat brain. They could renovate the striatum. They could also ameliorate behavioral deficits after intrastriatal transplantation in dopamine-denervated rats. So we went ahead with the first transplantations on November 10 and December 8, 1987, in 2 patients. We identified the ventral mesencephalon, they made a cell suspension, and which was implanted into the stratum of 2 patients unilaterally. And here you can see D-Day, the first transplantations we did with human fetal tissue. On the left, you can see Patrick Brundin with clothes suitable for dissecting the tissue. Anders Björklund is checking the viability of the cells. And then from the surgery room, Stig Renkrona is putting in the tissue, and also Håkan Widner, who was responsible for the immunosuppression, is present. Of course, this was a major event for us, and we were very, very excited about what was going to happen. And what we had decided was that we should not reveal anything during the first 6 months. Patients were monitored very carefully. We had a PET scanning at 6 months to see if growths had survived or not. And then I was going to present the data on an international symposium on Parkinson's disease in Jerusalem in June 1988. And the meeting approached, the media interest was very, very high. But unfortunately, what we saw was only minor improvement, and there was no detectable change in fluorodopa PET, even if we later found that there was a small dopaminergic growth on autopsy 21 years later in the first patient. So I was standing there. The media interest was very high. There was a film crew from the BBC, so my presentation showing now, once again, in principle, lack of improvement. So that presentation was filmed, and I was, of course, very disappointed once again. But this was not the time to give up. And I remember a quotation from Winston Churchill saying that success consists of going from failure to failure without loss of enthusiasm. I think that was perfect at this moment. But we had to improve the transplantation procedure, and Patrick Brundin found in his preclinical studies that the size of the implantation instrument was important. And we also realized that we had to have more implantation sites in the putamen in order to cover the entire structure. So therefore we went ahead with transplantation in 2 more patients in April and May 1989. And I remember that summer because I was evaluating the patients once a week clinically, and then once a month with an L-DOPA test. And after 3 months in patient number 3, one could clearly notice that there was an improvement in his clinical condition. His mobility on the side of the body, contralateral to growth, was faster, rigidity was less. His condition in the morning before the first L-DOPA dose was improved, and also the L-DOPA dose, if in the test, had a longer effect. So after 5 months, we decided that we should see whether this improvement was due to a surviving dopaminergic growth. And we decided to perform a PET scan. And this time, Anders and I, together with the patient, we went to London. And I remember this, that already at the time of the scanning, when we were sitting there looking at the computer, there was evidence that that there was a surviving growth. And I remember how excited I was. Would it really be possible? I didn't believe that it was true. And when I came home to my wife, and she asked me how was it, how did it go? And I said that, well, I didn't know, maybe I didn't. But after a few days, it was confirmed that there was actually a surviving growth in the putamen. And this finding that it is possible to put neurons into the adult human brain, a brain with a chronic neurodegenerative disorder, and get survival of these cells, and that they can actually influence the function of the host brain, that was the most exciting finding during my entire scientific career. And of course, this also, when our paper was published in Science in 1990, it created an enormous media interest, and it was on CNN. There were many, both national and international papers published reporting of this finding. This is our more local paper, Lund Scientists Succeeded. This is more like some kind of Hamlet version of the story. And then also there were reports in Wall Street Journal, New York Times, Washington Post. Since then, there has been 4 major milestones on the journey, on my scientific journey. And the first one is that growth can survive, that they can release dopamine, and that they could, in some patients, be associated with major clinical improvement despite an ongoing disease process. Also, especially after the negative sham surgery control trials, to demonstrate that dopaminergic denervation pattern outside grafted areas determine the functional outcome after transplantation. The finding that Lewy bodies can be present in a small fraction of grafted cells more than a decade after transplantation, which has triggered a completely new research field in Parkinson's disease. And finally, that serotonergic neurons in the graft are important for the adverse effects that we and several others reported after transplantation, the graft-induced dyskinesias. And I just want to mention one of these milestones, because it was on one of the occasions where Peter Hagel, who was then my PhD student, he is now a professor, and we went with 2 patients for PET scanning in London. And then Paola Piccini told us that she had a colleague who had been able to monitor and quantify dopamine release in vivo in a normal subject who had been playing a video game, and do that with PET scanning. And the method is that you use raclopride as a tracer. It binds to dopamine D2 receptors, but it is displaced by endogenous dopamine. So high binding indicates low dopamine release, and low binding, high dopamine release. Of course, immediately this triggered an idea that would it be possible to quantify dopamine release from grafted dopaminergic neurons? So we planned for this study. We had a quite perfect patient, a unique patient for this purpose, who had been grafted only on one side of the brain, and the other side could then be used as a control. I must admit that I was not very optimistic. I was excited, but I was not very optimistic. I didn't think it would work. But the patient was very enthusiastic. He had done very, very well after transplantation and been able to stop L-DOPA medication. So this patient and we went to London. We had dinner in a nice restaurant after 10 years after his transplantation. Before transplantation, he could hardly move without L-DOPA. Now he hadn't taken L-DOPA for many years. So we did the PET scanning, one scan with saline, one scan with amphetamine to look at the drug-induced dopamine release from the grafts. And then we went home. And I didn't hear anything from Paula, and I was absolutely convinced that it had failed. So after a couple of weeks, I called Paula and asked what happened. And she told me everything went exactly as predicted. It was the ideal scenario, which means that the patient in the fluorodopa scan, he had a normal fluorodopa uptake, Whereas with the raclopride, both with placebo and he had on the grafted side, similar to a normal subject. On the non-grafted side, there was a high binding, which indicates that there was very little dopamine. And then you could, by using amphetamine, you could release dopamine and quantify that. Nothing happened on the non-grafted side. So in this patient with a major clinical improvement, the graft had also restored fluorodopa uptake as a measure of dopaminergic innervation and dopamine release to normal levels. And our paper was directly published in Nature Neuroscience, and we even got this cover on the issue. Yesterday, where I was unfortunately not able to participate, you heard about the current status of grafting and also with fetal cells, and that there are two sham surgery control studies which have not shown any significant clinical benefit. And I will not discuss that here, but I would just want to mention to you what I think are the best evidence from the open label trials that this strategy may work. And these are two patients who have been followed, and they were selected in London and have been followed in London. And this patient, he was diagnosed in 1983. He was grafted bilaterally in the stratum in 1993 and 1994. And 4 years after transplantation, he could withdraw his L-DOPA medication, dopaminergic medication, completely. This patient was diagnosed in 1985. He was grafted bilaterally in 1996, and also during the 5th postoperative year, he could remove all his dopaminergic medication. And there are 2 points I want to make. One point is that at 16 and 13 years after transplantation, 26 and 24 years after diagnosis, and about 10 years after withdrawal of L-DOPA medication, they have a UPDRS motor score around 10. And gradually, their condition or their fluorodopa uptake has become normal, and they also have a normal dopamine release in the stratum, in the putamen. What I think one can conclude from the cell transplantation field in Parkinson's disease is that there has been a steady scientific progress in this field since 1985. There is proof of principle that neuronal replacement can work in the diseased human brain. Grafts can re-innervate denervated areas, they can become functionally integrated, they can release transmitter, and they can restore function in the diseased 50, 60 years old patient's brain. But as I'm sure you heard yesterday also, current cell transplantation procedures do not provide clinically competitive treatment. So why do I think that we are living in an extremely exciting time now for cell therapy research? Well, now we know that neurons for transplantation can be generated from different sources of stem cells and reprogrammed cells. You heard that yesterday. Not just dopaminergic neurons, but also other subtype-specific neurons. And also that we have neural stem cells in our brain, both in the normal and in the diseased brain. And finally, that there is now also evidence that you can generate neurons directly from glial cells and other cells in vivo in the brain by in vivo reprogramming. Yesterday you heard that dopaminergic neurons and also other types of neurons which may be important for Parkinson's and other diseases. You can get dopaminergic neurons from neural stem cells. You can get them from pluripotent cells, generated from blastocysts, human embryonic stem cells, or fibroblasts. And you can also get dopamine neurons directly from fibroblasts, and they can be used for transplantation. But we also have neural stem cells in our adult brains. And new neurons are continuously generated in these areas. These are the neural stem cells around the ventricle, in the lateral ventricle, and then we have also in the dentate gyrus, giving rise to granule cells and important for learning and memory. And we showed several years ago in rodents that these neural stem cells, they can respond to an injury, to a disease, by, for example, in stroke, where there is a big lesion in the striatum and the overlying cortex, they start to proliferate, they form new neurons, neuroblasts, which migrate towards the injury and then differentiate to mature neurons. And this potential self-repair mechanism was recently, the interest was recently stimulated a lot by the finding from Jonas Frisen's group using carbon-14 dating that actually in the adult human brain, there is in the subventricular zone, there is a proliferation of cells, of neural stem cells, and they give rise to neuroblasts which enter the striatum where they differentiate to striatal neurons. And interestingly, this mechanism is suppressed in patients with Huntington's disease. And finally, and this is of course very far from the clinic, that we know now that glial cells can be directly reprogrammed to functional neurons in vivo in the adult rodent brain. And what scientists have done is to use a viral vector expressing a transcription factor that has been injected into the brain and been able to convert astrocytes to functional neurons. And this has also in vitro been shown to be able to convert a human glial cells, an astrocyte, to a functional neurons. What will come out of this, we don't know, but we now have many, many tools to continue to develop this field, which makes me extremely excited. What I wanted to say at the end is that I hope I have illustrated for you that there is no fast track for cell therapy in this type of disorders. But with the rapidly increasing knowledge about brain plasticity and cell plasticity, I'm convinced that we will be able to develop clinically competitive cell therapies in the future. But finally, if you ask me, how far have we reached? Where are you now on this development? How far have you reached? I would like to answer by using another quotation from Winston Churchill. I think we can say that this is not the end. It is not even the beginning of the end, but it is perhaps the end of the beginning. The journey I've been telling you about has not been possible without a lot of excellent scientists, and I want to thank all of them for having contributed to this journey. Many of them are my very close personal friends also since many years. So thank you all very much.
I just want to mention that Dr. Jankovic showed a picture of the founders of the society, and all but one of them are in the audience today, and it must be very gratifying to see how far this field and our society has come these last 30 years. Congratulations to our junior awardees and to Dr. Jankovic and Lindvall for fantastic contributions this morning, and that will conclude this morning's session.